So, on that note, I would like to introduce David Rubinow as our first speaker. David is without doubt, and I know that there are protégés of his in the audience, but he's without doubt a giant in this field. He has been there since the beginning of this realization of the importance of this disorder. He is the Asad Maimandi Professor and Chair of the Department of Psychiatry at the University of North Carolina at Chapel Hill. Prior to that, he was the Clinical Director at the National Institutes of Mental Health, where he was the founding chief of the Behavioral Endocrinology branch. At UNC, he established a women's mood disorders program that is the only such program that is NIH supported for training. And I believe you have the only inpatient maternal mental health unit in the country. That's amazing. It was a great pleasure that I invite David Rubinow to discuss, give us an overview of maternal mental health issues, how they might differ from mental health issues outside of pregnancy. We're really excited to have you here.
So while Shadon makes me functional here, that and my coffee, they wouldn't let me bring my IV setup in here, so I have to drink it like this. First, I do want to say what a privilege it is to be a part of this, because I think you all represent really the sort of the statement of, necessary statement of values that we all should represent, which is the health of children and the health of mothers. And we seem to have really lost our way, certainly as a country. And this arguably is the most important preventable public health problem. Just how do we take care of our mother's health, and how will that impact the offspring? So actually, Adre asked me to, as way of an overture here, to define and to sort of provide a way of understanding a couple of very narrow topics like stress and depression and pregnancy. Actually, what I've decided to do is just to summarize the entire topic on the first slide. And it says, admittedly, it's a complex situation fraught with dangerous implications from conflicting interpretations of the facts and the pressure of historical entities, but I'm sure we could come up with something if only I could remember what it was we were talking about. Now, what I will be talking about primarily are concepts that I think you will hear repeatedly over the course of the next 2 days. And it's this: how do we really think about how the fetal environment gets translated into different outcomes for the child? And the first and most important is context, because physiology is entirely context dependent. Your genetic background, your sex, certainly, but also timing. Sort of when something occurs, and what else is occurring at that time. It's not just what occurs, but it's when it occurs in development. And that raises these issues of critical periods and plasticity, which is to say that a stimulus that occurs at a particular period in development, perinatally, during puberty, results in a reprogramming of the brain so that it'll respond very differently to the same stimulus, as opposed to that stimulus, that event occurring outside of those critical windows. It is worth saying that these periods of plasticity are also being addressed therapeutically, because there are ways of reopening them, like with amblyopia, as a way of really trying to repair disorders of development. Adaptation, resilience, and vulnerability. I mean, we as creatures are always attempting to adapt to our environment. And what are the factors that result in someone responding to environmental stimuli by being resilient, able to manage it, or by being susceptible to the adverse effects of a bad environment? Sensitivity is one that I'll focus on a lot, because most of these reproductive-related mood disorders represent a differential sensitivity to the same stimulus. So different people respond differently to the same stimulus, which after all is exactly what we deal with every day when we're dealing with mental disorders. The idea of networks, which are responsible, these neural networks responsible for providing most of the kinds of emergent cognitive functions that we have. And then finally, the concept of state, which is a different way of thinking about mental disorders from the usual menu of options that we throw together in all these different ways to come up with diagnoses. Now, when we're talking about stress, there is no single definition, but I would define, just for straw man's sake, a stress as a stimulus that requires an adaptation to or anticipation of or preparation for an environmental stimulus, right? I mean, we all sort of know what we mean by stress, and it results in our experiencing something, and it results in our having to generate a response to that stimulus that either prepares us for the future or that allows us to manage it at the time. And the response is usually experienced as some kind of arousal, either this sort of locus coeruleus sympathoadrenal system, or what people focus on a lot, the hypothalamic pituitary adrenal axis that generates glucocorticoids, the autonomic nervous system, your heart rate goes up, your blood pressure goes up. And then there are changes in the activity and transcription of very specific brain regions like the limbic system, which you all know about, and the amygdala in particular, which assigns affective valence to stimuli and helps us to remember them. But when it comes to the factors that determine the response to stress, they are myriad. So the nature of the stimulus will determine what the response is, right? It isn't a stress as it matters whether it's a psychological stress or a physiological stress. It matters whether it's conditioned or just experienced. It matters whether it's repeated, how long it occurs, whether it's occurring rapidly, whether it's occurring often. The timing of it was mentioned. But timing I wanted to focus on just for a moment, because when we're thinking about the fetal brain, we have to think, as Dr. Weinberger and Bale will talk about, and actually Dr. Epperson, the genetic symphony that occurs over the course of development really occurs in these so-called sort of gene network waves, which is to say, it's not like genes get turned on during development and they all just happen to stay on. You know, genes have very different trajectories. Some of them will go on and then not be on anymore. Some of them will have waves. Some of them will be inhibited. Some of them will be constitutively active. So a stressor occurring at one particular point in time is not just going to affect whatever genes are operative at that time, but since these genes all talk to each other and interact each other's function, they will have downstream effects on genes that come online later in life. So again, knowing when something occurs, but also what the downstream properties are, will determine what the stress response is. Antecedent history is really critical, and sort of the best way of demonstrating that is from the work of Bruce McEwen, who looks at, now these are genes that are activated or decreased in the hippocampus following a forced swim test that you put rats in a bowl of water and they don't like it. But notice that if you just give them cortisol, which, right, that's the stress hormone, you see almost an entirely different set of genes that are activated, very little overlap. And if you expose them to a chronic restraint stress as opposed to an acute forced swim stress, again, you got a whole different set of genes. Further, if you look at the genes that are increased by a forced swim test, the genes that are activated by that same forced swim test following a 21-day period of chronic restraint stress, a whole bunch of different genes. And even more to the point, if you then let the animal recover for 21 days, you see an entirely different set of genes. What that means then is that once, in this case, the animal is stressed, its response later to a stressor will be entirely different. And further, although most of the genes get turned on and then they subsequently go off, some of the genes get turned on and stay on, and some of the genes get shut off and stay off. So it's not like once we've gotten through the stressor, everything's fine. So what happens? So the past is prologue to the future in terms of our response to stressors. And then finally, gene by environment. Well, I should mention perception, but I'm not going to. It should be obvious, but the learned helplessness studies really showed that the response to an uncontrollable stress is very, very different from the response to the same stress when it is controllable. Some really elegant studies. But it's all about this interaction between our environment and our genes. And Thomas Boyce and others observed something really interesting that was not intuitive. They focused on what they called reactive alleles, that is, these genes that are very responsive to the environment. And so this is a summary of a whole bunch of different studies, but if you take somebody, a child or a rhesus macaque, who is very susceptible to, very reactive to the environment, you can measure that with autonomic activity, you can measure with immune response, and you put these highly reactive kids in a terrible environment, a high-stress environment, they develop all kinds of medical illnesses, behavioral problems. Well, that's not surprising, right? They're reactive and they respond to an adverse environment. On the other hand, which is amazing, these same children in a nurturing environment actually do better than the non-reactive children. So these children who... really doesn't matter whether they're in a bad environment, good environment. They were referred to as the dandelions. We can grow anywhere, doesn't matter. These are the orchids. And so it really is quite remarkable because it suggests that this reactivity can result in a beneficial response to a beneficial environment, but also an adverse response to an adverse environment. So putting this all together in a way that is just overwhelmingly complex, because that's what it is, these events early in life, such as prior stressors, duration, intensity, and so forth, interact with genetic factors to create, if you will, different set points in these regulatory systems like stress, like the hormonal system or transcriptional changes, which then result in different adaptational positions on the part of our physiologic systems, different set points for our stress hormones, neurotransmitters, neuropeptides, which then ultimately can result in either resilience or invulnerability. Now, what I'm going to be focusing on are these stress hormones. I'm going to focus on the endocrine system for the rest of this presentation. And part of the reason for doing that is that the endocrine system is really a grand conductor. It's an orchestrator of physiologic events. It does it in the long term by changing every element of the response system, but it also does it acutely. So the first sort of demonstration of that, it isn't really the first, but you got to start somewhere, is with Hans Selye, who in this article in 1946 talked about the general adaptation syndrome. And what he was calling attention to is the fact that there are these physiologic responses to stressful events, to a lot of different stressful events. And one of the characteristic responses was this activation of the endocrine system, which he thought was going to be responsible for many of the pathophysiologic changes that people experience under conditions of stress. And then the second really interesting demonstration of the power of hormones in regulating the physiologic response was from W.C. Young's lab 13 years later in this very famous classic article by Charles Phoenix, in which they administered an injection of testosterone to a guinea pig prenatally, and it turned out that ever after changed the response of the brain, so that as an adult, the females who were exposed to testosterone in utero do not respond to estrogen with normal kinds of maternal behaviors. And in fact, when they were exposed to androgens, they acted like males. They tried to mount the other females. They're very aggressive. So a very early exposure during a critical period forever after programmed the sensitivity and a different sensitivity to subsequent exposure to reproductive steroids. This was called an organizational effect. You're organizing the brain as opposed to activating a particular physiologic response. So the question is, well, how do they do it? How do these steroids produce these effects? And in fact, this is really staring into the jaws of biological diversity because the complexity of our physiologic response is beautifully illustrated by steroids. So this is cholesterol. This is the mother of all steroids. And once cholesterol is taken up by the cell and goes into the mitochondria, this 6-carbon side chain is cleaved. I don't know how to make this not. I'm going to seize, actually. I'm going to need Andre's help here. Oh, there, that's better. Okay. It produces a compound called pregnenolone, and then pregnenolone gives rise to all the biologically active steroids, the estrogens, androgens, glucocorticoids, mineralocorticoids. And to follow the rest of the talk, it's really critically important that you remember every one of these structures because the point is really that the way in which these steroids act, what their effects will be, is determined by how they're metabolized. You see, they all look sort of alike, right? So if you take testosterone, you break this double bond, you've got dihydrotestosterone, which is a very, very, very powerful androgen. If you use the same enzyme and add another one that reduces this ketone group, you have a neurosteroid that acts really acutely at the cell membrane. If you add an enzyme called aromatase, you create estradiol, and then it acts through its estrogen receptor system. And in fact, in the male rat, this conversion of testosterone to estradiol happens in the nerve endings, and the estradiol is released into the synapse in association with male sexual behavior. So acting an awful lot like what we think of as a neurotransmitter. And these steroid compounds all are part of this family, this nuclear receptor family. And the receptors for each of these, they're all organized in fairly the same fashion. So you can see glucocorticoid receptors, androgen receptors, 2 different types of estrogen receptors, alpha and beta, which are coded for on different chromosomes, 2 different types of progesterone receptor. And because they're all set up sort of the same way with the hormones binding in this ligand binding domain, there's also a lot of promiscuity of hormones across all these different receptors, so you get a lot of off-target effects. So that can create unwanted and unpredictable effects of these hormones. Now, the way that they work is that the hormones are lipids. They enter the cell. They bind to a receptor. In this case, the estrogen receptor goes to the DNA, binds a hormone response element in the DNA, and then generates just gazillions of different proteins, right? I mean, the messenger RNA gets translated into different proteins. And these are proteins for neurotransmitter receptors, but also the proteins that are responsible for translation of all of the proteins in the cell, all 3 polymerases which are responsible for generating messenger RNA, transfer RNA, and ribosomal RNA. They're all regulated by estrogen. So estrogen prepares the cell for subsequent transcription of messages and translation of proteins. Now, it's a tiny bit more complicated than that. And this is just to point out that there are lots of different ways in which a hormone binding a receptor can generate its effects. So I just showed you one. A hormone binds the receptor, goes to the DNA. That's one way. Another is a hormone binds the receptor, and then it's so-called tethered to other proteins that can then activate genes that don't have these classical hormone response elements. That's another way. A third way is actually not just with these intracellular, intracytoplasmic receptors, but actual membrane receptors. And this has been a hot area of investigation over the last 15 years because on the membrane, there's estrogen receptor alpha, estrogen receptor beta. There's a G protein called GPER1. Estrogen can bind with proteins called covalins, and in such a way it can regulate metabotropic glutamate receptors. In other words, it can produce glutamate activity in the absence of glutamate. And these membrane effects are critically important for things like social behavior and reward in the brain. And then additionally, the estrogen receptor, which is one of the oldest receptors that are known phylogenetically. It is very, very ancient. It existed way before estrogen came into evolution. And so these receptors are activated by a whole variety of different signaling factors like neurotransmitters like dopamine, growth factors, and so forth. But what I want to focus on are these co-regulators, because this helps to explain why people have different responses to different hormones. And these co-regulators are both activators and inhibitors. They're what determine what happens when a hormone binds to a receptor. And so this is what an estrogen receptor looks like, and these blue balls here are estrogen in the ligand binding domain. And when a hormone binds a receptor, it changes the shape of the receptor. It twists it like a Lego. And in this case, what it does is it flips this part of the receptor, opens a coactivator binding groove, and that's where these proteins then bind. The coactivators then bring in all of their friends. They go to the DNA, they open up the DNA, they modify it, and then they transcribe it. Conversely, this is something that's binding the receptor, estrogen receptor. It's called a SERM, or selective estrogen receptor modulator. And this SERM flips it this way, twists it differently, blocks this coactivator binding groove, and now the coactivators can't bind, but the co-inhibitors can, shuts the gene off. So, number one, when you're thinking about hormonal actions, it's about shape. It's about how these proteins interact with each other that then results in their being enzymatically modified by other proteins. It's this incredible dance of proteins. The second thing is it's much more important than affinity. You don't know what happens with a receptor as a consequence of its affinity for a particular hormone, because it depends on how the shape gets modified. So, I'm killing you with all this basic science. I apologize. No, I'm almost done with it. I'm almost done. What I'm trying to point out again is, we think very on one hand, we think simplistically about biology, and it's really, 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 really complicated. On the other hand, it's really cool because it can explain why we have these very, very different responses to ostensibly the same stimuli. So you get all these different levels of hormones and receptors. It's all sources of variability. What I want to focus on here, you're going to hear a lot about epigenesis, so I'm not even going to talk about that at all, but that's incredibly cool in terms of understanding how the environment gets translated into a different biological response that can be enduring for the rest of the life of the individual. But I want to talk about one last element of this, which is really arcane, but it's this post-translational modification, which means after a protein is created, you can add all kinds of enzymatic groups to it. You can add an acetyl group or a methyl group. You can add a protein that's going to degrade it. You can add a phosphorus. And it turns out that that's what determines what happens when a hormone binds a receptor. So this is one such coactivator, steroid receptor coactivator 3. You can see, actually, that maybe steroids will put a phosphorus group on here. And if it does that, it twists the coactivator in a way that it now recruits all these different enzymes that go to the DNA and elicit one kind of transcriptional response. On the other hand, a growth factor puts a different phosphorus group on it. That results in the recruitment of different kinds of coactivators and a whole different genetic program. All right, so now just to make this ridiculous point even more ridiculous, there are at least 8 different sites on each of these co-regulators in which you can make anywhere between 6 and 8 different enzymatic modifications, and there are 350 of the co-regulators. So what it results in, just put a number on that, is 10 to the 13th different possible functional outcomes as a result of a hormone binding a receptor. All right, so take home. Structure is function. Not all hormone preparations are the same. The effects of hormones are everywhere. They're tissue specific and they're context dependent. All right, lovely. So what does that have to do with depression? All right. Well, one way of answering that question is that reproductive hormones touch every system that has been implicated as potentially physiologically relevant in depression. And I've just managed to do something. I don't know what I did. Oh, well, okay. I talked about the regulation of the synthetic and metabolic enzymes for all of the neurotransmitters as well as their receptors. The stress axis is critically modulated by estrogen. There's an estrogen receptor response element on the CRF, which is the head of the stress response. These hormones, particularly estrogen and progesterone, are neuroprotective. They regulate how myelin gets laid down on neurons. They protect cells against all kinds of assaults like ischemia and glutamate excess. They regulate mitochondria respiratory efficiency, which translates into the inhibition of these so-called reactive oxygen species that go around and wind up destroying proteins in our body and leading to cancer and other great things. Regulates adaptor proteins, co-regulators. But I want to focus on this, which is network dysregulation. And sort of how do networks lead to behavior? Because we're going to be thinking about this across all psychiatric disorders. So how does the brain work, right? I mean, that's what we need to start with right there. So with apologies to Audrey for the way in which I'm going to construct this. But neurons, the nerve cells in our brains, are all about signal processing, right? I mean, they turn on or they turn off. They fire or they don't fire. They get a whole bunch of different stimuli, and then they decide whether they're going to fire or not. And then neurons can begin to cluster into these networks, which means the different brain regions can dynamically group together to perform certain kinds of functions, like paying attention or like, thinking about how you were embarrassed by, your cousin at the last time you had Thanksgiving. Those are all performed by certain and different kinds of assemblies of these regions. And then this is what's critical, is that the synchronization of the firing of the neurons, the timing of the firing, is what conveys meaning to the rest of the brain, right? And that shouldn't be surprising because we're incredibly frequency-dependent organisms, We see things, light is frequency-dependent, color is frequency-dependent, sound is frequency-dependent. So if I go, da-da-da-da, what song is that? Nobody knows. How about if I go, da-da-da-da, right? Okay, there you go. Ah, da-da-da-da, la-da-da-da-da-da-da. Okay, so what happened? The pitch frequency, the inter-pitch interval, suddenly conveyed meaning to you. You not only recognize the song, you could tell me who did it, you know what country he comes from, you know that he got deaf when he got old. So it's frequency. That's how we respond to the world. And when nerve cells fire together, they convey important information to the rest of the brain. So how does this synchronization occur? And is this going to work? Yes, okay. So imagine that nerve cells are firing at the same frequency, okay? And these metronomes are like nerve cells. These are all at the same frequency, but they're all out of phase with each other, right? So now if you take these metronomes and you put them on these these soda cans, what happens is they can slightly influence each other's activity. Small perturbations in their activity allow them to begin to influence each other so that now they all become synchronized. This is in effect what happens when a critical mass of neurons is firing. It changes the extracellular environment. And it changes the extracellular environment, and that just perturbs the other nerves enough. Why is it important that they're synchronized? Because when they're synchronized, they are maximally receptive to incoming stimuli. It amplifies the response. And hence, these signals can emerge from the noise that's the background in the brain. And, all right, stop already. Now, it turns out that this process of firing together also changes the structure of the neurons, and estrogen plays a major role in that because within minutes of application of neurons to estrogen, you see these new synapses being formed. And these synapses will disappear if there's nothing that follows. In other words, there's nothing to be learned, so it goes away. But if there's any important incoming information that's frequency dependent, they become stabilized synapses. So estrogen actually changes not only the synchronicity of the firing of the brain, changes the wiring of the brain. And you can see from this work from Bruce McEwen's lab, again, these are the synapses in these hippocampal dendrites. This is what they look like during proestrus when you have high levels of estrogen and progesterone. And then during estrus, you see what happens. You don't need statistics to see that estrogen is really dramatically altering the brain. So reproductive steroids regulate neuronal signaling. All right, still, what has this got to do with depression? And here's where I think our thinking has been really terribly misguided. Because depression is not a hardware problem. There isn't a lesion or a gene that causes depression. It's a software problem. It's a failure of orchestration and integration across neural networks that results in an adaptive failure on the part of somebody with depression. And so is there any evidence that there isn't a problem with the orchestration of these networks in depression? The answer is absolutely. You see problems with the so-called default mode network, in which you're sort of thinking about things, daydreaming about them, not focused, social cognition network, reward circuitry, affective regulation, and so forth. But then the question is, all right, well, are these networks regulated by reproductive steroids? And the answer is, of course they are. This is studies done with Peter Schmidt and Karen Berman, looking at 3 different conditions. One using this drug called leuprolide, which shuts off the reproductive endocrine axis, in essence creates a reversible medical menopause. And then with substitution or with addition, supplementation of estrogen alone or progesterone alone. And you see that these critical regions of the default mode network are highly activated when estrogen is present, but not at all when estrogen is not present. How about the reward circuitry? Reward circuitry is arguably the most important when thinking about depression, because what's the default? What is the despair that somebody with depression experiences? It's not only that they can't change, it's that they can't experience anything as being rewarding. And if you can't experience anything as being rewarded, what are you going to be motivated to do? Nothing, right? Not eating, not drinking, not sex, not seeing. None of it matters. And you can't even make decisions. And you can imagine that if you felt for the rest of your life you would experience nothing as being rewarding, it's very easy to imagine why people become suicidal. So Jenna McHenry, who was a postdoc working with me and with Garrett Stuber, did a series of studies and identified an estrogen-responsive reward circuit in the brain. So she did an optogenetic study in which you take a light-activated ion channel rhodopsin, and you transfect it into this neurotensin neurons in the medial preoptic area, which goes to the stride, actually the VTA, into the nucleus accumbens to generate reward experience. And then she hooked the if I can get this, yeah. And then she hooked put in a fiber optic cable, and then allowed the mouse to stimulate herself, right, with this light. When the light comes on, it opens up the ion channels in these particular cells and activates them. And she did that in different points in the estrous cycle. And what you see, if I can see, here we go. So light comes on for 3 seconds when she nose pokes this lever. Now she's going to go away, but she's going to be right back because that really felt good. And so if you'll notice here, during the high estrogen phase of the estrous cycle, she will poke this 1,500 times over the space of half an hour. This is a highly, highly, highly rewarding experience relative to estrus itself, which is only a day later. So the reward circuitry is certainly active. And additionally, what she was able to show using in vivo 2-photon calcium imaging is she was able to demonstrate that specific cells respond to the stimulus, and they respond to a sexual stimulus, but not to an appetite stimulus. So these cells are really very specific in terms of their reward properties. Okay, no, wrong way. Telling me here. All right. And the other element of this, which I think is critically important, is the phenomenologic confusion. And this is what Andre was talking about. You know, we've got all these different psychiatric disorders, and they've got all these symptoms, and we say that this constitutes a disorder like depression, right? Well, there are 595 ways of diagnosing depression when you put the symptoms together, right? And yet we're saying this is all the same syndrome. Is it really the same syndrome? Someone, an adolescent girl gets depressed when her boyfriend breaks up with her, as opposed to a mother who becomes acutely depressed in the postpartum, as opposed to, I'm really way behind, I realize. Anyway, the point that I want to make is that instead of thinking about these disorders as collections of symptoms, we should be thinking about them as state-related disorders. So what do I mean by a state? A state is a self-organized ensemble of different kinds of ideas and affects, memories, ways of thinking about the world and other people. It's a program for interpreting the world. So you might be getting ready to see a patient and you say, I just got to see this borderline patient, and he's just going to say, I don't know what I'm doing, and, you know, my mother's right, I should have been a surgeon. What am I doing this for? And I don't like whom I'm living with, and DC, I mean, my God, you can't drive anywhere anymore. And then something great will happen. You have a great meal with somebody, or go on vacation, or go to a sporting event. You go, hey, I have done better with this patient than all of her preceding therapists. I am a budding luminary in the treatment of borderline personality disorder. How fabulous it is to be a mental health worker where you can really improve the quality of people's lives, not putting zippers in people. And it's just, and DC is so, we won the World Series. And you call that a change in perspective. That's a change in state. And we all look to change our states, right? I mean, that's why you go for a run in the morning, or you have a beer at the end of the day, sex, drugs, rock and roll. It's all ways of changing your state. So if we think about reproductive disorders that way, looking here at premenstrual mood disorder, here you see what's really interesting is not just that the symptoms that are increasing in the during the luteal phase, but look how rapidly they disappear. There's a switch that gets thrown that puts someone into a different state. So that suggests that we can really manipulate behavioral steroids to determine whether they play a role in reproductive related mood disorders, and if they do, why do they only do it in some women? So finally, that brings us to perinatal mood disorders. And I'm going to run through all this anyway, so I'm not going to do that on that summary slide. So what are they? You've heard from Andre. These are very common. About 500,000 women a year in this country develop perinatal depressive disorders. They are the most common unrecognized complication of the perinatal period. It's more, additionally, they're associated with tremendous morbidity, devastating consequences for everybody in the family, a low maternal weight gain, preterm birth, small for gestational age offspring, impaired bonding between the mother and the infant, which predicts learning and behavioral disorders for the infant down the line, increased risk of suicide, according to the Confidential Inquiry into Maternal Health in the United Kingdom, the major cause of maternal death in the year following delivery. More than sepsis, more than hemorrhage, it's suicide consequent to perinatal depression. And, of course, an increased risk of infanticide, particularly with postpartum psychosis. And yet, there's no routine screening, no practice guidelines. So, I'm stuck again. Okay. So what are the symptoms? Unlike classical depression, where you think more about sadness, they're really more anxiety, irritability is a prominent symptom, agitation, anger, feeling overwhelmed, feeling you can't care for the child, and then shame about it and guilt. impaired bonding, I mentioned, impaired concentration, profound rumination, hypervigilance about the baby, intrusive thoughts. The timing of it I've summarized here. And note that about more than a third actually develop the depression during pregnancy, so it's not really appropriate to call it postpartum depression. About a third to 40% in the first 4 weeks, about another 20% in the next 8 weeks, and then people will then say another 10% even out, although, thinking about a postpartum depression occurring a year after onset just doesn't make any sense to me. So we have to then recognize that these disorders we're calling perinatal disorders are really a diverse group of disorders, right? All kinds of different symptoms you can put together. 2 women with postpartum depression or postnatal depression may not have any symptoms at all in common, yet we call them the same thing. And the timing is also different. But in terms of the risk, it's pretty clear that the past predicts the future. That if you have a past psychiatric history, particularly a history of bipolar disorder, eating disorder, even major depression, you're at high risk for developing a postpartum or a perinatal depression. But it is very responsive to the social environment because other risk factors of poor support, marital stressors, sleep disturbance. I mean, the most important thing we can do for someone with a perinatal depression or to prevent it is to make sure that they sleep, preserve sleep at all costs. Depression during pregnancy, obviously, substance abuse, and trauma is huge because trauma is endemic and it is a major factor. So, teleologically, we can say, well, is there any advantage to mood regulation? I mean, why does this even occur? And the answer is, how can you expect otherwise? Because really, reproductive steroids have to be able to change motivational state. Otherwise, mothers aren't going to take care of their kids. As Michael Newman demonstrated with rats, that exposure to reproductive steroids changes the brain's response to an infant's cry. And these elements of reward and decision-making are all critically modulated by reproductive steroids. So if we're but do the steroids actually cause perinatal depression? So one might look to them because if you see what happens to the hormonal changes during pregnancy, estrogen goes up 100-fold and then bang, it just drops way off, which gave rise to this notion of estrogen withdrawal. So the way that we decided to study the role of reproductive steroids in perinatal depression was by using a compound called leuprolide acetate. So this is the reproductive steroid axis. This part of the brain, hypothalamus, controls the pituitary, releases hormones, controls the ovaries to secrete estrogen and progesterone. And leuprolide actually shuts off this whole axis, creates a medical menopause. And so then what Mickey Bloch did is he took women who had a history of postpartum depression who were well, and he gave them Lupron to shut off their ovaries and then exposed them to high-dose estrogen and progesterone and then blindly withdrew it. And this was all blinded. So it's a scaled-down study of the puerperium. And what he found is that during hormone add-back and during withdrawal, you saw a big increase in depression, but it disappeared within a couple of months of this initial withdrawal. But note that women who had no history of perimenopause, of postpartum depression, those women had absolutely no mood perturbation at all in response to this stimulus, this endocrine system. So it looks like the hormones are responsible in this group, but not in this group. Right? And recently, Crystal Schiller performed the same study, but she also did brain imaging at baseline and then brain imaging during withdrawal. And she replicated Mickey's findings, again, finding that the group with a history of postpartum depression got very symptomatic during hormone add-back and hormone withdrawal, whereas the controls did not. You see this across a number of symptoms. And further, what she found is that if you broke the group of those with a history of postpartum depression down, or perinatal depression, you saw 2/3 of them responded exactly the way I just described, that is, they were hormone sensitive, but about a third of them didn't show any change at all. So that means even in a group of women who have a history of perinatal depression, some of them will be hormone sensitive and some of them won't, which is exactly what Mickey found originally. So then Crystal used a monetary incentive test to activate parts of the brain to see if she could distinguish differences in brain response in those individuals who are hormone sensitive from those who were not hormone sensitive. And what she found in one of the three areas that she looked, this area called the putamen, she found that in the controls, the hormone insensitive women, you saw sort of a little bit of an activation during hormone withdrawal. You saw the exact opposite. You saw a decrease in those who were hormone sensitive. And in the whole brain analysis, you saw this exact same phenomenon, this opposite effect in the putamen, the cingulate, the insula. These areas are critically involved in regulation of affect. In fact, the putamen and insula are part of what's called the hate circuit. This is, a suggestion that the brains of women who are hormone sensitive respond differently to exposure to reproductive steroids. Now, estrogen and progesterone aren't the only things that change, right? Because remember, the effect of the hormones depends on how it's metabolized. And one metabolic product of progesterone is this thing called allopregnenolone. And allopregnenolone is a neurosteroid that binds to the membrane of the cell, of the neurons, and acutely changes its activity. Allo is like the anti-stress hormone. It shuts off the HPA axis activity. It is a major GABAergic activator. And in fact, oh God, this is one of these slides that comes on in portions, which I hate. All right. So these GABA receptors, the allo binds particularly to GABA receptors that have subunits called delta subunits and alpha 4 subunits. And they exist outside the synapse. And their job in brain activity is to cool off neuronal activity. Right? So this sets the tone, so-called tonic inhibition in the brain. So the idea would be that you have fluctuations in these neurosteroids that lead to changes in GABAergic activity, that lead to dysregulated neural networks, that lead to a postpartum depressive episode. So one way of testing this was to actually administer allopregnenolone to pregnant women, which Samantha Meltzer-Brody did in our unit that Audrey spoke about. And in the phase 2A study, within 12 hours of putting in an IV and administering allopregnenolone, the depression was gone. These are hospitalized depressed women. And in the larger phase 3 study that showed, this was about 300 women, and this is also, Neil Epperson is also part of the study. You saw a superior response and a superior remission rate in those getting allopregnenolone as opposed to those who didn't. And note that even though the infusion was for 60 hours, the effects lasted for 30 days. Which brings us, now I have about 4 minutes. You're not counting, okay. I'm almost done, actually. It brings us to this question, which is sort of what causes susceptibility? Why do some women get these disorders and other women not get these disorders, right? It's a critical question. One of the best answers to this comes from work done by Jamie McGuire, who created these two really wonderful models for postpartum depression, one of which involves removing the delta subunit from or knocking out the delta subunit gene from mice. And the delta subunit, remember, is part of that GABA receptor that the allopregnenolone binds. And what she found when she did this, these mice look perfectly normal until they deliver. And once they deliver, they look depressed. That is, that postpartum, they're anhedonic, if you will. They don't want sugar anymore. They're immobile in the forced swim test. Their HPA axis is dramatically upregulated, and they kill their infants. Their pups, I should say. And so they kill their pups, although that can be reversed if they're administered a version of allopregnenolone. So it does suggest, and she has another model to suggest the same thing. So note This is a genetic abnormality that presents with no symptoms except in the context of a change in reproductive state. Now, when we talk about genes in humans, it's mostly sort of as illustrated here. You know, Jimmy's sixth generation pain in the ass. We think about these, you know, horrible traits of people that make them unpleasant to be around. But we wanted to look at this in relation to these reproductive mood disorders. So the first one that we looked at, I am almost done, is premenstrual dysphoric disorder. So these are weekly mood ratings. You see the increase in symptoms in the fourth week. If you shut off the reproductive axis with Lupron, the symptoms all go away. Now you take these women, and now what happens when you blindly add back estrogen and progesterone, and you see they get depressed. But the exact same hormone manipulation in women with no history of PMDD, absolutely no perturbation of mood at all. So you can't say, well, it's a steroid that causes it in everybody. It doesn't. It's an interaction between the genetic, well, presumably genetic background, and although it doesn't have to be, and the endocrine state. So the way in which Peter Schmidt and Dave Goldman and Lima Duby and I decided to look at this was to create a lymphoblastoid cell culture line. So you can take lymphocytes, turn them into lymphoblasts, and then compare the women who had hormone-sensitive PMDD. Right? We showed in the Lupron study that when you shut off their reproductive steroids, their syndrome went away. You add back the steroids, comes back, and looked at their genetic transcription under baseline conditions and when exposed to estrogen or progesterone in vitro, in a cell, in a dish, right? Because we're trying to see whether there is any evidence, any cellular evidence of this differential sensitivity to hormones. And among the genes found was this very interesting family. This was also done in a replication sample, incidentally. Upregulation of almost all of these transcripts, about 5 of them significantly so. And this is a gene family that is an epigenetic modulator. It's a so-called polychromosome repressor complex, but it methylates genes to shut many of these genes off. And note that the response to, particularly to progesterone, was very different in the cells of women with PMDD compared to the controls. So this same approach, this cellular model of sensitivity, has also been performed, this is my last slide, in women with a history of perinatal depression. And these are really hot off the press kind of data. But these 3 major genes, which are a translational regulator under conditions of stress, actin cytoskeletal regulator that influences synapse development, and a calcium signaling regulator, all of these were depressed in the cells from women with PMDD, with perinatal depression. So, in sports, first New York Rangers lost last night 3-2, but claimed victory anyway. This is really complicated stuff. I mean, seriously. But the fact is that if we employ the right conceptual models instead of trying to put people into cubby holes, and if we really use these conceptual models in concert with these wonderful tools that we have available now for understanding the brain, and we recognize that there is not one size that fits all, we're going to be, like T. Albert Fencer, in a much better place to be able to accomplish exactly what Andre was talking about initially, to understand how the fetal environment can actually serve as either a healthful or an adverse context for the development of children, which is, after all, the health of which we're actually all trying to preserve. So thank you very much for your attention. Oh, good. I can repeat it. I can hear.
Hello. Yeah, I don't think it works, but so my question
So the question is, did we examine menstrual cycle characteristics and a past history of responses to birth control? We certainly got histories of responses to birth control, although when you're dealing with these hormone-sensitive disorders, particularly with I can't things anymore, with help me out, Neil. Premenstrual, yeah, no, I know, really. My estrogen levels have dropped, obviously. Premenstrual dysphoric disorder. History is only of limited value. We didn't find any differences in terms of response to oral contraceptives. That is, people saying I had a terrible response or I had a good response, which fits the literature. If you look in the literature, it's sort of a third, and a third. People say, third say I had no response at all, third say it made me worse, a third say it made me better. So that wasn't a predictor. In terms of menstrual cycle irregularity, we constrained the baseline menstrual cycle limits as part of our entry criteria. So if somebody had a vastly irregular cycle, we just couldn't study them because we would have no way of knowing whether the ostensible response to Lupron, for example, was simply a function of the fact that they had an inovulatory cycle. So I don't know whether, if you looked at women who had really irregular cycles, whether that would, tell you something about relative hormone sensitivity. Don't know the answer. Sure. No, please.
During these studies, were there or have there been in other studies a look at the results with the differentiation of the ratio balances, not just the progesterone and the estrogen, but testosterone in particular, especially for those women who are in fact hormone sensitive?
So, I guess everybody heard the question. It's a complicated question. So one is sort of hormonal balance and the other is, and related, is sort of testosterone. First of all, there is a lot of, for lack of a better word, hype about hormonal balance. You see that there are many people who set up shops and say, I'm going to get your salivary porcelain levels and I'm going to balance everything and it's all going to, and then I'm going to I'm going to send you to a compounding pharmacist to create exactly the hormone that I want. Now, am I saying that people don't respond to that or that hormonal balance isn't conceptually, if not practically, important? And I'm not saying either of those two things. I am saying, however, that there's no science behind many of the claims associated with hormonal balance. Having said that, testosterone in women is, I think, an emerging area of importance. The Endocrine Society just published a position statement on that. They said that testosterone was only indicated for treatment of hypoactive sexual desire syndrome. But I can tell you from my own experience and that of and of course I'm forgetting his name. My estrogen, please. No. That would be terrible. I am blocking in his name. It doesn't matter. Anyway, the person in the UK who is the editor of Climacteric, and anyway, what they say is they say that there are absolutely women, particularly perimenopausal women, for whom testosterone has absolute psychotropic and cognitive effects. Well, we don't know who those people are, and there's a real problem in the perimenopausal women because most of the assay methods for measuring testosterone that will be obtained in many of the laboratories are simply not adequate in perimenopausal women because the levels are at the lower limit of detectability of the assay. So I think that we need those studies. We really need good clinical trials to see whether there are ways of being able to improve someone's sense of well-being, their cognition, their mood, with very, very specific manipulations, be it administration of testosterone, which, by the way, in women is far more plentiful and far more abundant than estradiol is, right? I mean, testosterone is not only a source of estradiol, but, it's an order of magnitude greater in its concentration. So, of state. I think there absolutely should be studies looking at these hormones as regulators of state, because the animal models, again, are very compelling in suggesting that hormones are critically involved in dominant states that become imperturbable. If you remove estrogen, you wind up inhibiting GABAergic activity in the basolateral amygdala, and you get an excitatory state that just persists and it won't stop. And it results in the animals being incredibly sensitive to stressors. And the only way of stopping, a way of stopping it is by raising the estrogen levels again. So I think what you're suggesting is something that I think is critically important. That instead of focusing on the symptomatic state itself, we need to understand that switch process. We need to understand how people get into those states, how they get out of those states. And what is it that allows many of us to get into one of those states and we feel terrible and then something nice happens and we feel okay, whereas somebody else winds up becoming unable to get out of that state. And that gets at this notion of susceptibility and vulnerability. It's a lack of ability to restore homeostasis. In a way, it's what Bruce McEwen would talk about as sort of an allosteric set point, meaning your physiology gets moved to a new place and you can't get out of it. And there are great seizure models for that as well. So I think hormones do everything, but I think particularly the role of testosterone in women, on one hand, absolutely needs to be studied. On the other hand, I heard testimony from somebody just last week that said that he's giving testosterone to reproductively active women because he thinks that it prevents breast cancer, and with emphasis on he thinks. That's just really bad form. Yes?
Just along those lines, is there a difference in symptomatology in women who have a male fetus, a boy, versus a girl?
I don't know. Neil, do you know the answer to that? So there are differences. Yeah. And in this whole area of again, thinking about the animal studies, sort of the sex of the proximate rat pup influencing the subsequent development, which apparently it does, which gets at you're going to hear a lot about placental biology. And so I think that following Dr. Monk's point and Dr. Epperson's, that this is where you see the potential for the sex of the fetus really potentially, at least having an impact on the fetus, if not on the mother. Thank you again very much.